All right, so we all remember learning about DNA in school, right? That classic double helix, the blueprint for life. But it turns out that's really just the first chapter of a much bigger story. There's this whole other set of hidden rules that decide how our genes actually work. And they play out on every scale, from entire populations down to these tiny little chemical tags sitting right on your DNA. So let's dive in and explore some of these hidden rules.
I mean, it seems like they should, doesn't it? If a trait is kind of rare or if it's recessive, you'd think it would eventually just get, well, washed out of the population. You'd expect a dominant gene for, say, black fur to eventually make all the hamsters black. But that's not how genetics works. And that brings us to our first big idea.
You see, to answer that question, we can't just look at a single family tree. We need to zoom way out and look at the genetics of a whole group of animals. This is a field called population genetics. It's all about seeing the entire forest, not just one or two trees.
Okay, so imagine you could take every single gene from every single individual in a population, let's say all the moths in a forest, and throw them all into one giant collection. That collection, that's the gene pool. And the different versions of a gene, which we call alleles, they're like different colored marbles in that bag. You might have marbles for black wings and marbles for white wings, all mixed together.
So here's the kicker. Evolution, when you get right down to the genetic level, is really just a change in how common each of those marble colors is over time. If the percentage of black-winged marbles goes up from one generation to the next, hey, that population is evolving. But what if it doesn't change? Well, to understand change, scientists first had to imagine a world with no change. It's a kind of baseline, a perfect, idealized scenario where that gene pool just stays exactly the same generation after generation. And this idea has a name, the Hardy-Weinberg equilibrium. Now, the most important part of this whole principle is that tiny fine print at the end, in the absence of other evolutionary influences. See, in the real world, things like natural selection, mutations, migration, they are always happening. So this principle isn't really describing reality. It's giving us a perfect mathematical model that we can measure the messy real world against. Okay, let's make this real with a classic example. Let's say we have a hamster gene pool, where 80% of the alleles for coat color are for dominant black fur, and just 20% are for recessive gray fur. Now, with black being dominant, it really feels like the gray hamsters are fighting a losing battle, right? Let's see what the math actually says. So, how do we predict what happens next? We use the Hardy-Weinberg equation. Here, we call the frequency of the dominant black allele, P, which is 0.8, and the frequency of the recessive gray allele, Q, is 0.2. This grid, which you might remember as a Punnett square, shows us all the possible combinations. The odds of a baby hamster getting two black alleles, that's BB, is P squared, or 0.64. And the chance of getting two gray alleles, BB, is Q squared, which comes out to just 0.04. And here are those results laid out in a chart. You can see that in the next generation, 64% of the hamsters are BB and have black fur. And only a tiny 4% are BB with gray fur. But look at that middle bar. A full 32% of the population is BB. They look black, but they are secretly carrying that recessive gray gene. And that is our answer. The gray hamsters don't disappear at all. Why? Because that recessive allele, B, is protected. It hides out in that huge 32% of the population that are carriers. The math proves it. As long as things stay stable, the frequency of that gray allele will just hang out at 20% in the gene pool forever. Okay, so that's the math that governs the big picture. But what about the genes inside you as an individual? Well, the story gets even cooler because our DNA isn't just some static book that gets read from cover to cover. There's this whole other control layer sitting on top of it, deciding which pages get read and when. And this brings us to the really interesting difference between genetics and epigenetics. Think of your genetic code, your DNA, as the fixed architectural blueprint for a house. It is what it is. Epigenetics, on the other hand, is like the contractor. It's the set of instructions that tells the builders which parts of the blueprint to focus on, which to ignore. It's the software that runs on the hardware of your DNA. So, to put it formally, epigenetics is the study of changes in how genes get used without actually changing the DNA code itself. The name literally means on top of genetics. These changes are kind of like putting sticky notes on your DNA that say, read here or skip this part. Or you can think of them like dimmer switches that can turn a gene's activity way up or dial it way down.
So how in the world does this actually work? Well, a perfect example is something called DNA methylation. It's a really simple but incredibly powerful process. A tiny chemical tag, called a methyl group, just attaches itself to a gene. And that little tag acts like a physical roadblock. It literally stops the cell's machinery from being able to read that gene. In short, it flicks the gene to the off position. And this is not some minor process. It's absolutely essential for building a body. It's how a cell in your arm knows to become the skin cell and not a brain cell.
Okay, for our last big idea, we're going to zoom all the way in, I mean deep inside ourselves, to the little power factories, where we find a completely separate second set of genetic instructions with its own totally unique rules of inheritance. That's right, tucked away inside the mitochondria, you remember, the powerhouses of the cell, is a second tiny genome. We call it mitochondrial DNA, or mtDNA for short, and it is completely separate from the main blueprint, the nuclear DNA that's safely locked away in the cell's nucleus. And when you put the two side by side, the differences are just stark. Your main nuclear DNA over on the left, you get from both parents, we all know that. But your mitochondrial DNA, get this, it comes only from your mother. It's also incredibly tiny, just 37 genes versus more than 20,000, and it's unbelievably efficient with almost no wasted space. It is playing by a completely different rulebook. Which, of course, brings up a massive question. Why? Why only from the mother? If we get half our main DNA from our dad, why does literally none of his mitochondrial DNA make the cut? Well, the answer seems to come down to two main things. First, it's just a numbers game. An egg cell is absolutely packed with mitochondria, maybe 100,000 of them. A sperm cell? It has only about 100. But even more than that, any of the father's mitochondria that do manage to sneak into the egg during fertilization are actively hunted down and destroyed by the egg's internal machinery. And the implication of this is, well, it's pretty profound. Think about it. Since you got your mtDNA from your mother, who got it from her mother, who got it from her mother, and on, it forms this direct, unbroken genetic line connecting you to your maternal ancestors, stretching back thousands and thousands of years. So, from the vast mathematics of an entire gene pool down to the little epigenetic software that runs on our genes, it's clear the rules of life are way more complex and interesting than we thought. Which kind of leaves us with one final thought. If our environment, our lifestyle, our experiences can actually leave these epigenetic marks, those little sticky notes on our DNA, it makes you wonder, right? What story is our life writing for the next generation? What instructions are we leaving behind?